So let's get started by introducing our first presenter, Dr. Douglas Walsh, who is the Research Director of the Environmental and Agricultural Entomology Lab located at the IAREC in Prosser. Doug?
Great, thank you, Judy. I think I've popped my screen up here, and I hope all of you have watched the riveting 32-minute presentation I created with the assistance of the AV group here. So anyway, moving on, I'll try to be brief here. This is the team that I've been working with. A number of them I've worked with for a number of years. We're pleased that Justin has joined at the University of Idaho. He'll develop into being a very key collaborator for me. And then here located at the Prosser Research Station, we have the addition of Kayla. Sally and Dan are sort of the anchor upon which I build my program. Sally produces much of the high quality documents that are produced by our group. And then the key to what Sally does for us as well is we just turned in what's referred to as our stakeholder relevancy statement yesterday to compete for another specialty crop research initiative grant. And I think all of us on the team can commend Sally for the outstanding job that she does in putting these research proposals together on our behalf. So I had 8 mite management objectives planned for 2020. They were a combination of research, service, and extension. These were our objectives for this past year. Basically running field efficacy trials, conducting bioassays with candidate alternative caricides. We've been working with establishing mite colonies and breed resistance. We had a little bit of a setback this year where our colonies did die, but the California strawberry growers were more than willing to send us some mite populations up from California. And as I've said before, anything that we've observed in the Pacific Northwest pales in comparison to what we've observed in California strawberry plantations. I think this objective here, E, is one that Justin will hopefully talk about, but I think we'll make inroads in developing this sensitive and rapid cost-effective method to predict multiple caricide resistance on a portable platform. Currently, we have research going on to see how winter mortality affects mite populations. And then we're going to screen the advanced cycle germplasm. So I'll be working with Kayla towards looking at the potential resistance in the male germplasm we have at the research station. And we sort of dropped off on this objective just due to the COVID issues of us not being able to put more than one person in a car at a time this past year. Now, this here is just sort of I'm gloating a little bit because Kunle, my former PhD student, just received this email yesterday. And so we've had a review paper accepted in the Journal of Pest Science, which is a fairly high impact journal in pest management. And in large part, I owe this, I owe this manuscript to the support that the Hop Research Council and the Washington Hop Commission have provided. And basically, much of the information here is derived from the hop system. And hops are the agro-ecosystem that we use as basically our model system. So this is a good thing for us, and this will be published fairly quickly. So it shows you sort of the science that we've put out. In 2021, in regards to the objectives I put forward, again, sort of the two objectives that are new for this coming year, again, are looking at the fitness cost associated with the caricide resistance. And then again, we'll be expanding our efforts with Kayla in terms of looking at the male germplasm as a potential source for mite tolerance. And again, I think that our project has benefited the hop industry. And again, we're excited to be doing this work with Kayla. As I've said before, the male germplasm has largely been ignored. We kind of put it in the corner and let it grow. We rarely sprayed it. Sometimes it even looked like a weed patch. And so I'm excited that maybe we can actually determine if mite resistance or susceptibility is a heritable trait, and then maybe that can be included in the breeding cycle. But moving forward, I have my second proposal here, and this is the integrated management of secondary pests on hops. And again, we've conducted most of this. We're looking for moths, we're controlling caterpillars, we're looking at potential controls for aphids. And then in terms of what we've done, in terms of what we'd like to expand upon, is this coming year, we've added a new objective. There's been interest by hop growers to see whether or not augmented releases of the predatory mites would somehow benefit and help reduce mite populations in hop yards biologically. And so we'll be working with some commercial insectaries to purchase and or get these mites donated to us and be testing these out in commercial hop yards. And with that, I'd like to thank the Hop Research Council and the Washington Hop Commission for the research dollars they do provide for us. And, you know, as I always say, the industry money is sort of the rock foundation upon which we can build our research programs and truly do make us competitive in other funding, mostly federal programs, and oftentimes, and here in Washington State, and state programs as well. And I'll get rid of my stop share, and I entertain any questions that anyone has.
Michelle, do we have any questions for Doug?
We have not had any come into the Q&A.
Okay.
But I'm certainly watching it. So if anyone does have a question, you're welcome to add that to the Q&A button at the bottom. I'll double check chat and make sure nothing came in that way. But
I either dazzled them with my brilliance or baffled them with you know what. So.
Well, and while Justin is speaking, some questions may come in, and we can have we can bring them up when he's done as well. Oh, we've got a question here. Looks like Doug from Joshua. It says, do you foresee predatory mites having a big role in the management of mites?
Well, certainly they already do. You know, here in Washington, in our hop yards, I'd say the predatory mites are probably the second most important mite predator. We really do see a lot more mite predation going on from our spider mite destroyer beetle. Stethurus is the generic name. And, you know, I've done, well, many years ago now, I worked with the California strawberry industry in doing mass releases of predatory mites in the strawberry system. And right now it is a sort of a tried and true method for controlling mites in that system, strictly because they burned out all their acaricides, you know, due to the amount of acaricides that they sprayed. I think they can play a role in hops. I think my fear is that the release rates that we would probably have to go up to truly get a measurable treatment effect will be cost prohibitive. But I think at this point, the industry has made it perfectly clear to me that it's something, an avenue that you'd like to have researched, and we'll be researching it this year.
Oh, all right, we've got a couple more questions. Doug, from Megan, are you going to be looking at new chemistries for aphid control? They are a bigger problem in organic production than mites, and imidacloprid is facing greater restrictions.
Well, the answer is yes. There are some new chemistries coming forward. For those of you that are familiar with the chemistry spirotetramat, it's registered on hops under the trade name Ultor. That was sort of the first insecticide in what's referred to as a lipid biosynthesis inhibitor. And they are now sort of becoming the new kid on the block. Gallon Chemical Company has new chemistry they're putting forward. And then Bayer Chemical Company, which was the original developer of spirotetramat, has sort of new and improved formulations in the pipeline. Now, in terms of controlling sucking insects, including aphids, organically, that's always a tough one. We've worked not in hops, but in wine grapes with wheat hoppers on a number of the like organic oil, rosemary oil products. Tetracurb is one of the trade names, with some limited success. Again, it's tough in with aphids and organic production. So there is some hope, and yes, we would plan on screening those sorts of materials.
All right, we've got another question from Grayson. What are your thoughts on efficacy of Savanto on aphid and mighty mint oil on spider mites?
Well, with Savanto, we've had very good results. Probably, I think the original work I did was probably 6 or 7 years ago. And again, it was a fairly effective aphicide in our hop trials. With the mighty mint oil, I presented results based on bioassay data that we did in the laboratory in 2019, and at the rates that were recommended of that product for use in recreational cannabis in our bioassay arenas in the Potter spray tower, we basically hammered the mites. Worked extremely well. When we dealt with the manufacturer of mighty mint oil, we sort of bounced around rates of how much would be affordable for hop growers, and we put that out, sort of reduced the rate from what a recreational cannabis grower might use to a rate that might be affordable, and we included that in our research trials last year. It really didn't perform that well under field conditions. So I think if you bumped up the rate, it would be effective, but I think it would be cost prohibitive unless you truly were getting a price premium for something like organic hops. In a conventional standing, I think most growers would probably wince if they saw the price tag.
Thanks, Doug. Another question from the audience. Are there any negatives to predatory mites? Any unknown side effects to other crops or habitat?
Absolutely not. The thing about predatory mites is I had, this will come off worse than I mean it to, but I used to have a strawberry farmer that used to say they're like farm workers. They're always going to go to where the pay is best. And so, what that means is you could release thousands of them in your hop yard, and if there's a peppermint field next to you that's full of mice, they're going to go there. They'll move on. So there's the predatory mites that we're working with this year are not obligate predators, which means that if they do kill off all the spider mites, they have alternate sources of food. In fact, some of the predatory mites actually feed on powdery mildew. Others will feed on pollen and things like that. There's specific predatory mites like the Persimmon species they use in California strawberries that are obligate predators. Once the prey is gone, then they either die off or move on. So we're trying these other predators that are not obligate predators in hops. So there's absolutely no negative consequence of having predatory mites in hops.
Okay, that looks like pretty good timing. That's concludes the questions for now, unless there are any others.
Great.
Okay.
Well, thank you.
Yeah, good timing.
Thank you, Doug. Appreciate it. Okay, so we're now going to have our next presenter in the entomology research section, and that is Dr. Justin Clements, who is the new assistant professor, extension specialist in entomology at the Parma Research Station in Idaho. And Justin just started this position early last year. So Justin, if you would take it away.
Perfect. I'm just going to share my screen real quick here. Put this up into... Does that look good?
Looks good.
Perfect. So, well, thank you very much. I'm very happy to be here to talk today. I will be talking about novel control of California Prienus beetle with entomopathogenic nematodes. It's a project that we submitted last year to HRC, and it's something that we're very excited to be working on. I unfortunately will not be talking about some of the work that Doug and I are doing with developing in-field assay for miticide resistance, and hopefully in the future, Doug and I can both talk about that. But sorry about that, Doug. For this presentation, though, as a new project, I wanted to give quite a bit of information about background of the system and the objectives for our project. So I'm going to talk about the life history of the California Prienus beetle, current management practices for this insect. Then I'll move on to entomopathogenic nematodes. I'm going to call them EPNs from now on. Talk about their life history, the biological requirements to use them in hop fields, and how they've been used in other pest systems. I'll then move into talking about the objectives for my HRC project and some preliminary data that we have generated so far. So the California Prienus beetle is a large insect. It ranges from, it's native to the Western United States. It ranges from central Mexico all the way up to Alaska, and it's sporadically located throughout the Pacific Northwest. This insect is a pest of 21 different plant species, from hops, grapes, and multiple different orchard trees. There's quite a few members actually of the Prionus family. They belong to the longhorn beetles, and they can be found throughout North America, Europe, and Asia. There's approximately 25 closely related species. However, the California Prienus beetle is the only Prienus species that is pest to hops in the Pacific Northwest. They have a long lifespan. They can live between 3 to 5 years. Most of this lifespan is spent as larvae in the soil, which is the pest stage of this insect. They have a relatively short lifespan as adults. They're only present for right around 2 to 3 weeks at the end of June and July. However, adults are highly fecundant, and females can lay between 150 to 200 eggs during their short lifespan. The eggs will be laid on the soil, then move into the soil, or the larvae will move into the soil where they'll feed on hop plants, hop roots. So again, the pest stage of this insect is the larval stage, and the damage is caused by larval feeding. What it does is it decreases nutritional uptake. It can cause water stress and reduce plant growth. Above ground, we see symptoms including the wilting of plants. We can see yellowing of vines. We can also see death of vines and the entire plant. These guys, unfortunately, can also introduce different fungal and bacterial pathogens into the root system. So they can be a significant problem in hop yards where they're located. So management around these insects relies on a few different techniques, including pre-planting fumigants. MOCAB EC is registered for control of Prienus beetles. However, it has a 90-day pre-harvest interval. Otherwise, you can remove infected plants or destroy fields that have high infectivity rates. There's different methods too of trapping insects. My predecessor, Jim Barber, did a lot of work on pheromone traps, and they did a great job. Trapping of these insects can be done using light traps and pheromone traps. However, this doesn't target the pest stage. This only targets the adult stage that's out in the environment for a relatively short period of time. And unfortunately, some of this technology has not been registered yet for pest control. So it's more of a monitoring of the adult stage of these insects. So we started to think about how we can add value to the hop system and how we could also control California Prienus beetles in a different manner. And actually this got brought up because we had a question from Diane Gooding regarding control of black vine weevil with nematodes. And I put her into contact with my boss, Dr. Edwin Lewis, up in main campus. And we started talking about nematode control of different soil dwelling insects. And we started talking a little bit more, and we started to put together some ideas on how we could actually control California Prienus beetles with nematodes. So these nematodes, EPNs, are going to be insect-specific parasites. which will predate and reproduce in the insect host. So these are going to be specific to insects, and you're not going to have to worry about these as far as damage to your hop plants. Death by these nematodes is actually caused by a mutualist, a bacterial mutualist that causes septicemia and kills the insect. These nematodes are going to be soil inhabiting. There are two major classes. However, there's a lot of different species within these families. They were first identified in the 17th century. However, work on nematodes and pest control really started in the 1920s and progressed a lot in the 1950s. Infections by these nematodes can take up to 3 weeks to kill insects. However, some nematode species can predate on insects in a relatively short manner and kill insects within 24 to 48 hours. I want to go into a little bit more detail just on how these nematodes actually kill and infest different soil-dwelling insects. So these nematodes can be naturally found within different fields, or they can be introduced. You can buy a lot of different nematode species from commercial vendors. What you're introducing is actually a juvenile into the field, and juveniles are mobile, these juveniles nematodes. They will seek out or ambush different soil-dwelling insects. They enter the insects through different openings the insect has, the mouth, anus, or spiracles. Once the insect enters into, once the nematode, enters the insect, it releases a bacteria. The bacteria is actually what's going to kill the insect. After the insect dies, the nematodes start to reproduce. Eventually, the insect will rupture and release more nematodes into the field, at which point these nematodes then can go seek out and infest other insects. So it's something that can be relatively sustainable. However, these are living organisms, so they do require certain environmental conditions. EPNs prefer sandy soil. However, they have been found in cultivatable fields, forests, grasslands, and deserts. They need a pH of between 4 and 8. They can survive generally between 0 and 40 C. Again, they're a living organism, so they can be killed through freezing, extreme hot temperatures, desiccation, and UV light. They need adequate soil. They also need adequate soil moisture for movement. And there's two different types of nematodes. There's ambushers and cruisers. They're literally what they say. So the ambushers will wait for a soil-dwelling insect to come upon them, where they'll be infested, and the cruisers actually go out and seek insects to infest. So EPNs have been used in pest management extensively, and what I have put up here are two different figures that look at EPNs in different systems, and it shows kind of the range of different insects that they have been able to infest and being used as control. I think the important thing, though, to get out of this figure is that not just one type of nematode can affect all different soil-dwelling insects. Nematodes will be, different species of nematodes will be specific to soil pests. So really what needs to be done is to figure out what nematode can infest the California Prunus beetle to have a system that is that can work in hop yards. So this kind of brings up to our proposal, what we proposed to HRC. The first thing was to determine optimal EPN species for California Prunus beetle control by laboratory and greenhouse assays. Next, we wanted to conduct field trials with EPN species that we determined as being able to control California Prunus beetles at Gooding Farms. And then we really want to take this information and establish different bulletins and guidelines for growers, because there are EPN species right now that can control some pests in hops, but however, that information is relatively limiting, and I think it just needs to be put out there in a bulletin that can be easy to use. So far what we have started to do is we've started to collect preliminary data and collect different California Prunus beetles. We collected beetles in May, August, and November at Gooding Farms. In total, we collected 24 larvae ranging in different size. They were driven up to our collaborators up in Moscow, Idaho, Dr. Lewis and Dr. Stevens for preliminary EPN screenings. They started screening these California Prenus beetles with one type of nematode that could also infest black vine weevils. It's Heterodibictus bacteriophora. Sorry, that was probably said pretty bad. As that can also infest black vine weevil. However, that was not able to infest California Prenus beetle. So now we've moved on to screening to different species, and that's going on right now. So we're going to screen these insects until we find a species that we can infest, and then we're going to take that EPNs out into the field. So really briefly in conclusion, EPNs have had a long history of successful control of multiple different soil-dwelling insects. I do think California Prenus beetle is a good target for EPN control. It has a long life history in the soil, and it's a soil-dwelling insect. What we really need to do is determine an optimal species and concentration to kill these insects. And if we can determine that, these would provide a biorational and a safe alternative to some of the pest management for CPB, and add another tool to growers' handbox to control these insects for sure. So with that, I just would like to have some acknowledgements. I'd like to thank the people that have worked so far on this project, including Dr. Lewis and Dr. Stevens. I'd like to thank Gooding Farms for all their help. Also my two techs, Maggie Highlight and Brenda Nelson. I'd like to thank the HRC. I also need to thank Doug, because Doug has been incredibly helpful with all sorts of questions in hops or any different growing system out in the Pacific Northwest. He's been a great asset and made my transition to the University of Idaho much easier. I need to also thank the funding through my startup and the Hops Research Council. So with that, I'm going to take myself out of sharing screen, and I can answer any questions.
Jill, have we got any questions for Justin?
We do, yes. Justin, from one of our attendees, the question is, how far are we from commercial availability? So
that's a great question. A lot of these different species are actually already commercially available, and a lot of different producers provide different EPN species. However, we don't know what species can infest California Prenus beetle. So if one of those commercial available species can, the answer is very, very quick, and there wouldn't need to be a registration on hops. We could just use that EPN species. So just determining the correct species is really what needs to be done, and to make sure that it works in the field.
Okay, great. Our next question is from Marissa, and it is, what does large-scale nematode release look like?
So nematode release can be done through drip irrigation, or it can actually be sprayed onto the field. And I was looking this up a little earlier, and you can put it on with very similar machinery as to different insecticide applications too. It needs to be done a little bit carefully, but it's an easy transition to administering nematodes into the field. You would have to be careful because some of the current insecticides and nematicides would kill these nematodes too, such as Mocab EC would kill the nematodes in the field. So it is something that has to be taken into consideration, but they can be administered in multiple different manners.
Okay, next question from Benjamin. Have you established thresholds for control of Prionis based on pheromone trapping?
That is a great question. That is a question that I am not completely sure about. The Prionis trapping with the pheromone trapped is more of monitoring the insects, and it was done predominantly by my predecessor, James Barber. So it's something that I can look into and talk to Jim about a little bit and get back to you. But I'm not quite sure. You probably can tell the abundance within the field, but again, you're bringing in those insects from a surrounding area. So it's going to be a little bit challenging for sure.
Can I butt in here?
Yep.
Jim Barber led the effort on that, and he did publish a paper about a year and a half ago that we can email to Judy, and she can make it available for you to read. The concern is that the California Prionis pheromone is not a straight chain hydrocarbon. And so the registrant right now is Pacific Biocontrol, and the Environmental Protection Agency would require Pacific Biocontrol to do a whole bunch of toxicity, environmental fate, all sorts of work to get pheromone registered as a pesticide. So that does not discourage you from basically surveying in a very intensive manner. But at this point, it's just not financially viable for Pacific Biocontrol to do all the work that EPA would require for them to register the pheromone as a pest control tool. So, like I said, I'll provide Judy with the manuscript that Jim had published.
Thanks, Doug. All right, next question from Diane. Can you talk about the dosing time in the season?
Yeah, so dosing for nematodes generally happens early in the season or quite late in the season. And it's also going to depend on what else you're putting into the field. So for black vine weevil, those applications are going to be put into the field mid-spring or late in the fall. So, and it depends again on the pest that you're trying to target, because again, you're looking for the soil dwelling part of the insect. So most insects will have a relatively short soil dwelling life stage. California Prionis beetle, however, has quite a long life stage in the soil. So dosing could be done at multiple time points throughout the year, and we can work on that too to figure out the optimal dosing for this insect.
Okay, another question for you, Justin. How prevalent is CPB, and what's the crop loss acreage look like?
That's a great question. It's something that I've been trying to determine too. So this insect is very sporadically located throughout the Pacific Northwest. It'll be present in different growing regions. It is a pest down in southern Idaho, probably more than other regions in the Pacific Northwest. Crop loss from this insect, it is a long growing insect, so it takes time. So I do believe you just see yield drags over time until you get just an unacceptable amount of harvest. However, I'm not 100% sure. It's something I'm still learning as being relatively new here. I guess that's probably the best way I can answer that. So.
All right, we have a minute left and 2 questions, one left for Justin and one for Doug. So last question for Justin is, what level of efficacy have nematodes had on other crops where it has been used and studied more extensively?
So nematodes can be incredibly effective, actually. There is always some concern with using a living organism. For black vine weevil control is great. They can be more expensive than different insecticide controls too. Nematodes would be considered a biorational, so safer alternative or kind of an organic pest control. So in those situations where people want to move that route, it is a nice alternative. They can be tricky to use, but when used right, they can be very, very efficient. So, and for this project, I partnered up with Dr. Edwin Lewis, who is an expert in nematology. And I think, using both of our skills, we can definitely determine the optimal concentration to kind of figure out what is needed to be put out into the field and optimize, this system so it would be very effective.
Thanks, Justin. And then that last question for Doug. Do you think it would be possible to rear predatory mites resistant to HOP-approved acaricides?
And the answer is most definitely yes. Sort of the sentinel work on this was done by a scientist named Marjorie Foy in California almonds. And she basically was successful in the almond system. The citrus industry from Florida basically snatched her away from the University of California, where she finished her career. exactly doing that for predatory mice in the citrus system. So absolutely.
Okay, Judy, that's the end of the questions for that session.
Okay, great. Thank you, Justin and Doug.